Great. All right, thank you, Martin. Our last speaker before lunch is Eugene Duff from the fMRI Center at Oxford. And he's talking about network modeling and fMRI analysis.
Thanks, Tor. So this talk is going to cover a lot of the topics and the methods that you've heard about already with Martin's talk. But I think we're going much more down into the trenches with this talk. I'm going to basically attempt to give a bit of a broad overview or a taxonomy of both of types of experiments that can be used to do make inferences about connectivity and networks from fMRI data, and then a similar sort of taxonomy of the analysis methods that are used to analyze these data sets. Causation is one thing we might want to infer regarding the brain networks that we are studying with fMRI, but there are many other aspects that we can also study, and I think part of the message of this talk is that the breadth of network and connectivity modeling methods are useful to provide a very broad overview of the extent of dynamics and of what's going on in the brain and the network, making a variety of inferences about these networks. Some inferences can be quite straightforward, like just characterizing which regions correlate in their activity, and then there are more challenging inferences like causality, which Martin talked about. And we often want to characterize how these networks might change. This is the real strength of network analysis with functional data, is that we want to really be able to characterize how networks might change across different conditions or across patient groups. So I'll start now just to cover a few types of experiments that get to connectivity using fMRI. And I'll start with probably not a standard type of connectivity analysis, but one where you actually are able to drive the brain. And this is a recent study by Petkov and others, where they were able to stimulate the temporal cortex in a variety of regions and then characterize how this induced activity particularly in the frontal cortices. And they found, as they stimulated across different parts of the temporal cortex, they found quite a surprising variation in the extent to which different brain regions would induce activity in the frontal cortex, and a pattern that really didn't match the known hierarchy of brain regions in the temporal cortex. And so this type of analysis is a very direct and easily interpretable connectivity analysis using fMRI, where we can make some reasonably strong inferences about what you might get close to making causal inferences about the influence of particular brain region of these temporal brain regions on other parts of the brain. So this is where the activity has been induced by some external probe, as well as microstimulation. There are a few studies using techniques such as TMS, which can allow similar types of analyses. But in general, we don't have this sort of luxury to be able to manipulate the brain in this way to provide very controlled changes in activity in particular regions and see how that influences other regions. And certainly the type of the microstimulation study, we're not going to be seeing too many more of those in the future from that lab, unfortunately. So a more standard way of sort of inducing activation and variations in activation, so we can track correlations in brain activity across brain regions, is to have a varying task in our experiment. This is a classic study of visual motion, first published by Buchel et al., where they looked at how different types of visual motion modulated, and they tried to model how connectivities were varied across different visual motion conditions. Other tasks can be less controlled, and for example, people have studied how the watching of a movie modulates connectivity relative to other conditions, or particular scenes induce connectivity patterns or correlations across brain regions. A related type of approach has used data from meta-analytic databases, such as the brain map database. And again here, the variation in activation has been driven by the different experimental paradigms in the data that's being used in the data set. And that has driven these correlations that are being observed and characterized. Alternatively, and perhaps very extensively, spontaneous activity, brain activity, can be used to assess or to realize and explore correlations between brain regions. This was first discovered quite early on in fMRI and has been extensively studied, characterizing these resting state networks. So here we don't have any external experimental manipulation that producing the activation and activity variation in our experiment. Another aspect of the experiments that study connectivity is whether there are some experimental variable that's been manipulated to vary connectivity. And in many cases we don't have any such experimental variable at all. We just want to characterize the connectivity observed in a single condition. For example, in the microstimulation condition, the connectivity was constant. It was just being observed by varying the location of the microstimulation. Resting state studies have done a lot of characterization of the properties of the resting state networks. And similarly, the original brain map analysis simply assessed the overall pattern of brain networks that could be realized through an analysis of the entire brain map meta-analytic database. Alternatively, of course, we can assess how different conditions may modulate connectivity, and this can be more difficult in some ways because we have to estimate multiple states of connectivity, but in some ways, in many cases, it can constrain our models, and so we can provide more tight inferences about what particular aspects of the network might be being modulated across these conditions. And of course, group differences is one form of this. Finally, and this is something that Martin touched on briefly, we can split up our connectivity studies into those that use deterministic models, where we have we basically assume that we're exploring our correlations in our brain activity, which are a direct deterministic response to external variables. For example, the manipulation of the activation with the microstimulator. And the classic dynamic causal modeling is a deterministic system. And certain analyses of the meta-analytic analyses can also provide, also involve this sort of deterministic modeling of the data. More common, I think, is stochastic or modeling or analyses that have a stochastic element, where we don't control, we don't have a controlled, we don't have a model, we have some sort of stochastic generation within the brain of the activity that is providing the correlations that we're observing. But this is not just in resting state type studies, but many analyses of data sets where there's a cognitive experimental manipulation. The actual analysis is still going to be sensitive to and still exploring the random fluctuations of activity that is associated with these manipulations. For example, most analysis of this movie isn't trying to explore exactly which brain regions activate to specific scenes, but it looks at overall patterns of correlation. So to formalize the challenge of connectivity analyses a bit more, many studies you can sort of consider at some level. We're exploring some sort of network matrix where we have a measure of connections from each brain region we're studying, and how some sort of measure of connectivity to all the other brain regions. And we want to analyze or make some inferences on how this underlying network is structured and how it might be changing across conditions. But we can make, as I said at the start, we can make various inferences regarding this underlying network. The simplest inference is simply, are there some sort of correlations between particular brain regions? In this case, we're not trying to make an inference about causality or particular directness of the connections in our brain, but simply we just want to characterize, are there general patterns of correlation in the brain networks that we're observing. A step beyond that is to try to infer the presence of direct connections and separate those from indirect connections. And finally, another approach that gets closer to looking for causality is looking for the direction of these connections. Does the brain region 1, does its activity induce activity in brain region 2, or vice versa? And in each case, we can characterize how these inferences might vary across conditions. So to make these types of inferences, we have a huge range of analysis methods at our disposal. And they vary in many aspects. I guess the most classic is this distinction between functional connectivity, which is simply trying to characterize general correlations in the network, and effective connectivity, where we're trying to make more specific inferences regarding direct connections and perhaps directed connections, and distinguishing those from correlations due to multiple steps of a network. And these analysis approaches can then be extended to higher order characterizations of the networks. For example, these graph theoretic measures, which you heard about earlier today, and can also involve, modeling can involve very much lower level models, or biophysical models of underlying neurons. So let's look a little bit more, or consider a little bit more, correlation as a tool for exploring connectivity and networks. It certainly does have some very strong advantages and strengths. It provides a very simple characterization and interpretable that is computationally relatively trivial. And because it's a pairwise measure, we can very straightforwardly do mapping analyses. And it is often also proven provide adequate information to be highly discriminative for pattern recognition approaches. And it is a tool that tends to be often a base for graph theoretic analyses. So its flexibility has really generated a wide range of analysis methods that can characterize the connectivity and correlation structure in the data in a wide number of ways. And this is, in general, very important to provide the analyst with a general view of the dynamics of the brain. This mapping ability has allowed us to characterize resting state networks and do these seed region analyses, which provide a whole brain perspective of the correlations in the brain networks. But it does have some limitations, and these have been very well characterised. It doesn't permit or reliably permit the type of inferences many people want to do regarding more detailed features of the brain networks. It also can be sensitive to differences or changes in signal to noise, signal and noise levels, and it doesn't estimate biophysical variables anyway, so it really is a very high level characterization that will identify effects that need to be studied in more detail. So if we want to ask these more detailed questions about that underlying network, we need to use other methods. And there's a number of approaches available, as we've said, and Martin touched on a number of these. Just to summarise briefly, one general class of these methods to try to get at in more detail inferences about directed connections and underlying networks are covariance-based. These are often entirely based or derived from the covariance matrix that we observed, and there's some sort, but they use the entire covariance matrix to make inferences about directed or undirected connections. Just to give some insight into how this might be done, you might consider two regions, B and C, changing across two conditions. And we see that their correlation increases. But many of these approaches will also take into account variance changes in the signals. And so if region B also shows this increase in variance, this may suggest in a simple structural equation model that the connection between going from C to B has become stronger, inducing a stronger presence of signal from C into B. And this is, which wouldn't be the case if the connection was in the other direction. These methods, one key feature of these methods is their dependence on all the region. For every connection you're characterizing, the connectivity measure is dependent on all the regions you have included in your modeling. And so if you leave out a region, your inferences are likely to be very different if you leave out an important region. Another approach to trying to characterize causality is looking at more other aspects of the distribution of the signal in different regions. This is an approach. Lingam is one such approach, which is related to ICA, which looks at the extent to which different brain region signals are Gaussian, with the inference being derived from the fact that if one region is receiving multiple inputs from independent sources, that the distribution of that of signal in that region will become more Gaussian. Finally, there's these lag-based approaches, like Granger mentioned earlier. These are dependent to a strong extent on differential, or on being able to make inferences about the timing of, relatively fine timing of activations in different brain regions. Trying to say that, because you're making inferences that activity prior to the activity in one brain region can be caused by a prior activity in another brain region. This approach can be problematic because of the uncertainties about the hemodynamic response. Finally, we move on to biophysical models of connectivity, such as dynamic causal modelling. These often combine multiple of the approaches that I've mentioned just now, and will also try to model underlying biophysical variables, such as the hemodynamic parameters and other neural parameters. These are, I guess, are probably the gold standard way of modelling connectivity in the brain, but they have some clear issues. One is that they're computationally challenging, and so you often have to have a relatively restricted number of brain regions in your model. And this will mean that it can be difficult to include all the regions that are relevant to the network, and also you may be missing large dynamics at a different scale. For example, the resting state network activity, which is a large-scale network activity, or even confounds need to be modelled in some way in these models. Our group a few years ago did a comparison of many of these methods I've mentioned, trying to explore which methods could reliably make inferences about the network structure. This paper used dynamic causal models, so it used a biophysical model to simulate a whole range of networks and network structures, and then explored how reliably different methods could reconstruct these networks. So they started with a general network structure and simulated data using DCM. And this simulated data had the full hemodynamics. It also explored aspects such as incorrectly defined regions of interest for some networks and other types of events you might be observing in standard analyses. And then it applied the result, analysed the resulting time series using a wide range of network analysis methods. The analysis, or the outcomes that are looking at is how reliably these methods identified direct connections and didn't falsely identify indirect connections or not connected regions. Also, for those methods that were able to make inferences about directionality, it looked at how reliably it got directionality right. Overall, the results from this work showed that in many of these methods did not perform well, and that it is a real challenge to make robust inferences of networks from realistic fMRI data. But it did show that correlation, and particularly partial correlation, was fairly reliable at identifying direct connections. Obviously, it can't identify the directionality of these connections, and in general, few methods reliably identify directionality. In particular, with the particular models used here, the Granger methods performed quite badly because there was due to the hemodynamic variation across regions, and also that if there were errors in your parcellation, this would also severely affect your modelling. So to go in a little bit more detail about partial correlation, which was mentioned by Martin briefly, and this is basically when we're looking at looking at a pair of regions, we regress out all the other signals in our network prior to looking at the correlations between these brain regions. If there's still some correlation remaining after doing this regression, it's assumed that a direct connection exists. This approach proved quite reliable in the network analysis, or in identifying directed connections. But there are some challenges with this approach. It is an estimation challenge because you have to regress out. If you have a large network, you have to regress out many node time series. In particular, if you get up to a large network, such as a 200 node network, you need to have many time points before you can reliably regress out, to reliably estimate these partial correlations. One way to ameliorate this is to use some regularization methods, such as Friedman's regularized inverse covariance, or to do some regularization by identifying a group mean across all your subjects. In some work we did a few years ago, we found that these regularization approaches for partial correlation further improved the estimation of the networks as measured by our ability to discriminate conditions, different steady state conditions from each other. And so when we use these inverse regularized partial correlation, we're able to more reliably distinguish conditions than with regular partial correlation or even correlation or any other connectivity measures. But it's not perfect. There are scenarios where partial correlation can give you incorrect inferences. For example, if you have two uncorrelated signals feeding into a third, when you perform a partial correlation, you can get a negative partial correlation between these two brain areas which are truly independent. And other approaches such as DCM should more reliably distinguish these scenarios. Finally, I'm just going to touch on the effects of signal and noise level changes on our connectivity analyses. The scenario you can consider here is what happens if we're comparing two conditions and we see a change in the noise levels. And this is not an unlikely scenario, both in group comparisons where we may have a disease condition or in different states where some sort of confound may be stronger in one rather than in the other. For standard regression analyses, this isn't necessarily an enormous problem because our estimates of the strength of an activation, while they may become less reliable, they won't necessarily become greatly different. However, if we're doing an analysis that uses correlation or some sort of other stochastic measure, it's much more difficult to distinguish the true signal from the noise. And so our measure of correlation or many of our measures of connectivity will change when there's a change in noise levels. This opens up our analyses to being much more sensitive to a wide range of confounds. And it's very important to be carefully characterizing these in connectivity analyses. It also makes these analyses sensitive to changes in signal levels. And these are often not considered in connectivity analyses in a comprehensive way. So if we see a change in the amplitude of signal, we'll also see a change in our measured correlation or other connectivity measures. Just to show this in more detail, if we observe a reduction in correlation between two regions, our inferences are entirely ambiguous as to whether this might be a reduction due to an increase in noise levels or a reduction due to an increase or a change in the amount of, a reduction in the amount of signal that we are observing in our time series. For example, the bold fluctuations may be just lower amplitude across the whole brain in a patient group. So these scenarios must be carefully distinguished, and they are going to affect most connectivity analyses. Even biophysical models that try to account for many different biophysical variables need to be very careful to include components and model variable noise across conditions. So rather than considering these network matrices as people often tend to focus on the off diagonals, we certainly need to also focus on the changes of the diagonals of our network matrices. So just to summarize, I think I've tried to show you how, I'll show you that fMRI can provide a wide range of insights into the connectivity and the patterns of covariances across brain regions and how they might vary across different conditions. And certainly the different methods that we have at our disposal do have strengths and weaknesses. It's dangerous to rely, for example, onto an approach that uses only a small number of brain regions that may miss some large-scale changes in our signal properties that are occurring across a much wider range of cortex. So it's certainly important, I think, to apply functional connectivity methods and then to identify the large-scale dynamics in our data sets, and then perhaps when we are happy that we've distinguished and pulled apart these components, apply effective connectivity models to make more specific inferences about our networks. And I think that's it.
Thanks, Eugene. I think, thank you to all of our morning speakers. It's been wonderful, educational. Just in case any of you might want lunch, now's the time. So we're going to have lunch, and we're going to come back at 1 o'clock. So we'll start again at 1.